Hi, I'm DeWald. I'm the Chancellor of Washington State University Health Sciences, Spokane. Thank you for joining us today. We are having a conversation with Dr. Senthil Natesan, who is an assistant professor in the College of Pharmacy and Pharmaceutical Sciences here at Washington State University. Senthil, if you would do us a favor, tell us a little bit about your educational background, where was home, and then what brought you to Washington State University?
Thank you. First of all, thanks for having me.
Great to have you here.
Yes. Actually, I'm a pharmacist, basically trained as a pharmacist, and then I got training into information technology. I did my master's in bioinformatics, and then I got an opportunity to do PhD in the North Dakota State University,
yes,
combining both pharmacy as well as information technology. And my PhD is on developing computational models to study the structure-activity relationship. Also, I developed several predictive models using surrogate systems in order to capture the interaction of drugs with the membrane, biomembranes. Currently, as an assistant professor here in WSU, my research focuses on understanding the biological process, complex biological process, especially that happens at the membrane interface using computational techniques.
Okay, and you've been here about 2 years, is that right?
Yes, since 2006, July.
Okay, and so your laboratory studies how drug molecules will interact with the surface of cells,
yes,
and the membrane bilayers on the cells.
Yes.
And so you're looking both how those drugs will interact with proteins,
yes,
that are there, or how they even integrate into and potentially move across the cellular membranes. Is that right?
Yes, absolutely. If you carefully look at the fraction of currently FDA-approved drugs, almost 50 to 60% of the drugs interact with the protein that are membrane-associated.
Okay.
For example, Gproteincoupled receptors, ion channels, and even membrane-associated enzymes.
Mm-hmm.
It's fascinating to know that why nature has selected membrane as a location for these physiologically important proteins, because as we can imagine, once you administer the drug molecules, the process by which drug molecules find their targets is kind of totally random.
Mm-hmm.
And this is done by three-dimensional random motion. Interestingly, these drug molecules can also partition into membrane, mm-hmm, and take a 2D walk. And researchers found that combining this 3D motion and a 2D lateral diffusion can potentially increase the probability of drug molecule finding the protein to 500 times.
And so the work that you're doing is very interesting because it's looking at the way that drugs interact with the proteins that are on the cell surface,
yes,
and then the membranes. So are you helping us to understand how to better design drugs?
Exactly. Actually, membrane can act as a kind of temporary depot, mm-hmm, from which drug molecule is slowly released.
Yes.
So a short-acting drug can be converted to a long-acting drug with the help of membrane. And for binding sites which are hidden in the transmembrane regions, essentially membrane can facilitate, they can arrange the drug molecule in a particular conformation which is very, very suitable for the ligand binding. Okay. And also these drug molecules can present at the vicinity of the membrane, so their association and dissociation could be facilitated by the membrane, resulting in a longer duration of action. Okay. So we do not have any clear-cut understanding of this association-dissociation process, which is called binding kinetics, how mm-hmm, fast the drug can go and bind to the target, and how slowly they can dissociate. This affects the efficacy. We are trying to understand how you can tweak the drug structure such that you can take advantage of this phenomena to improve the efficacy and safety.
So your computational training in your
Yes.
background allows you to build models for how a particular drug will interact with a protein.
Yes.
And kind of the binding sites on the protein and so on. How do you then take your computational predictions and then look at what happens with the cell? Do you follow up and you look to see what happens in a cell, or do you collaborate with others that do?
Absolutely. This is a great question, and I recently got funded by NIH a Small Business Technology Transfer Award. This is in collaboration with Dr. Michael Gibson in Pharmacotherapy Department. Dr. Gibson has spent around like 25 years in researching a particular genetic disorder called succinic semialdehyde dehydrogenase deficiency.
Wow.
These patients, especially most of them are children, they have a deficient enzyme which is essential to convert succinic semialdehyde to succinic acid. In the absence of this enzyme reaction, this succinic semialdehyde is converted to two other products, GABA and GABA hydroxybutyric acid, which results in so many developmental deficiency, epileptic seizure, and all these conditions. So we are developing a drug discovery program using in silico models. So we developed a pharmacophore model. This is nothing but trying to understand how this receptor would look like in a three-dimensional space, capturing those important interactions. And then we recently completed screening almost 40 million compounds.
Goodness.
And we arrived at top 25 compounds, cherry-picked. And currently this is under investigation by a company called Eurofin. They test on a rat cortical membrane using radioligand assay. And the compounds with the highest affinity will be probably undergo lead optimization process and could turn into a potential drug.
So that's interesting. That is looking at a particular disease that's caused by a particular change in a protein.
Yes.
And then you have, with your computational processes, designed a program so that you can optimize for the relationship between a drug, a potential drug, and its target. And then you're following up
with the additional experiments.
Okay.
Yes, and if we find that even like one or two molecules with micromolar or nanomolar affinity, that could be an excellent lead for further development in order to optimize for like drug-like properties, safety, and reduced toxicity, if any. So, as you know, a drug discovery process takes like 10 to 15 years and $1 billion. We would think our research would have some meaningful outcome if we can even reduce a little bit of the cost or time involved in this drug discovery process.
Yeah. So it's very interesting, and I like the sounds of what you're describing, because what you're doing is trying to shorten the timeline and reduce the cost using your computational approaches that inform then the drugs that might be developed.
Yes.
So as you're working, do you have particular target disease states or things that you are especially focused on with your work?
That is the beauty of my research. Even though we take some receptors or disease condition as a prototype, our research could be applied to any diseases, any disease models. By using this in our R15, actually I got funded R15, that is Academic Research Enhancement Award. That supports both undergraduate and graduate research. This is mainly for our membrane research. We propose to study Gproteincoupled receptors. These are ideal system because we have a very popular example of two drugs that are used in asthma conditions. These are like beta-adrenergic agonist, salmeterol and salbutamol. They both actually look very similar, except that the salmeterol, it has some extended tail. Salbutamol you need to administer every 2 to 3 hours, whereas salmeterol can last for more than 12 hours. And we found that in our recent simulation, the salmeterol goes and insert itself into the membrane, which could be possibly one of the reasons why it has a longer duration of action.
I see. So there it's a scenario where not only is a drug interacting with the protein that crosses the membrane, but it's also interacting directly with the membrane.
Yes. So the reason my graduate student, actually the first crystal structure of this beta-adrenergic receptor bound to salmeterol was published just 2 months ago.
Okay.
So we took this protein ligand complex and simulated the various different pathways, possible pathways of dissociation to see what happens. So in 7 out of 10 simulations, this molecule dissociates and then goes and inserts itself into the neighboring membrane environment, which could possibly be one of the reasons why this molecule has a longer duration of action. Because this same molecule is again available for rebinding to produce the biological effect.
Very interesting. So are you then, if you're looking at G-protein-coupled receptors, which are these transmembrane proteins found in almost all of our cells, are there particular disease states that you're especially focused on?
Currently, we are kind of interested in opioid receptor, because as you know, this is one of the most critical problem currently going on, this opioid overdose, and so many deaths due to this side effects of these opioids, especially respiratory depression and constipation. All this lead to a lot of casualties. What we are trying to do, we are trying to exploit this allosteric binding site in this opioid receptor. So these conventional opioids actually causes 2 different signaling pathways. One is essential to take care of pain, and the other one is causing all these side effects.
And the signaling pathways are when you have a molecule that binds to the receptor, and then it activates a series of different steps in the cell to maybe change gene expression or the metabolism of the cell.
Exactly.
Okay.
Yeah, so one of these pathway takes care of the pain, and the other pathway is like a reason for all these side effects.
Okay.
So we are planning to design a drug that could only activate only one pathway, which is called biased signaling. So we are trying to exploit this transmembrane exposed binding site, allosteric binding sites, to achieve this biased signaling.
So you're trying to modify the molecular interactions that lead to a lot of the whole body side effects that we experience sometimes.
Yes, so to reduce those side effects and only produce desired effects.
And you're doing that again with your computational modeling.
Exactly, and it needs to be obviously tested experimentally, but we are trying to kind of get those initial conformational changes which are signature for this activation of this particular signaling pathway using computer. Because as you can imagine, all these biological events takes place in a time scale of like microsecond, milliseconds, which are practically not tractable by the bench experiments. So we use a tool called molecular dynamics simulations, which are also called called as a computational microscope. So you can visually look at what are all the changes happening, conformational changes happening at the atomistic details.
Wow, So you go from the computational modeling clear to the examination at the submolecular level.
Yes.
So let me ask you a little bit. So you're working with Dr. Gibson,
Yes.
who's in the College of Pharmacy and Pharmaceutical Sciences.
Yes.
And you said that you are working also with pharmaceutical firms in some of the work at this point?
Yes, so that STTR grant, it is in collaboration with a company called Speragen Inc. Down in Texas.
Mm-hmm.
Actually, that company was founded by one of the parents whose 2 children are affected by this disease.
Okay.
So
your work really does span the, as you called it, in silico, where you then do the experimental, but it has a very practical side. You are already moving toward
absolutely.
the development of products that will help people who are confronted with these really terrible diseases.
Yes, because these diseases are like, it affects only very small population, and
Mm-hmm.
pharmaceutical industries are not interested in investing their resources.
So related to that, Dr. Natesan, as you look forward, what are areas, kind of what I would call scientific vistas that you and your team are interested in? What are you interested in focusing on in the future?
So I want to share a very fascinating fact here. We have made a phenomenal progress in computing in terms of both processing speed as well as the storage, memory, everything. I will give you an example to bring this into perspective. Actually, all of us are carrying a great supercomputer on our hand, which was used as a supercomputing device in 1980s. Actually, our smartphones are like as good as, even like more powerful than the supercomputing facility available in 1985 Cray.
Yes.
In spite of all this progress, as far as simulation is concerned, we will not be able to simulate any biological event that happens beyond this, like, microsecond level.
Okay.
But most of the important biological events, like a ligand molecule passing through the membrane, binding to the protein, as well as dissociation, this happens at a seconds to minutes time scale.
Okay.
So I believe that any progress that is made in quantum computing, and future has to tell us, like, how much it is possible to access this biological phenomena computationally. So I am looking forward for those days.
So you're excited about the advances in softwares and hardware?
Yes, absolutely.
As we move forward. Well, what I would say is thank you for the work that you do, that you're addressing not just what happens at the cellular level with drugs, but you're really thinking about the diseases that you can help design the drugs so that we can then intervene in effective ways for that. Are there any other things that you might want people to know about the work that you're doing right now?
Well, thank you so much for the public to support our research. We will definitely do our best to bring down the cost of the drugs, which is very important, and also to address some important, like untractable diseases like cancer. So we are working.
So I'll reflect back a thank you, Dr. Natesan, because sometimes we as scientists forget that it is every person in this nation that is supporting the work, and by funding the National Institutes of Health, who then fund the work that you do, they're saying that we appreciate and want you to do well. And I'll say thank you for being at Washington State University. And thank you for joining us today with Dr. Senthil Natesan.
Thank you so much for this opportunity, and thank you for supporting me in this university.
Great to have you here.
Thank you.
Thank you.